Hello, and welcome to this Nature.com custom webcast titled Identification of MRSA using 3-Colour Crystal Digital PCR. Today's webcast is sponsored by Stilla Technologies. Stilla Technologies is a Paris-based European biotechnology company that focuses on accelerating the development of next-generation tests by providing biologists with tools for high-resolution genetic analysis. Using breakthrough microfluidic technology, Stilla has developed the Naica system, an innovative digital PCR system compatible with multiple chemistries and equipped with 3-colour multiplexing capabilities. By encapsulating all steps for digital PCR in a single chip, the Naica system offers a fast and user-friendly solution, rendering digital PCR accessible to all. Stilla advises and supports its customers worldwide through its dynamic and multidisciplinary R&D team, with expertise spanning from microfluidics to chemistry to molecular biology and to computer science. We'll begin today's webcast with a presentation and end with a Q&A session where we will address the questions that have been submitted by you. To ask a question, just type your question in the Ask a Question box and press Submit at any point during the webcast and we will answer them today. It is now my pleasure to introduce today's speaker, Dr. Denise O'Sullivan, who is a senior researcher in the Cell and Molecular Biology team at LGC. Over to Denise.
Hello, my name is Denise O'Sullivan. I'm a senior researcher in the Molecular and Cell Biology team at LGC. I'm going to talk to you today about the identification of methicillin-resistant Staphylococcus aureus using a 3-colour crystal digital PCR. We work with many different commercial companies, so here I would just like to outline a disclaimer where certain equipment or instruments and materials which I will mention in this presentation, I am not implying recommendation or endorsing them, nor is it intended that they are the best available for that purpose. Now I would like to introduce some background about LGC. LGC is one of the UK's national measurement laboratories, and since 1988 it is the designated institute for chemical and biomeasurement. I work within the LGC Health Science and Innovation Division, where we perform measurement research, which is called metrology. We form part of the UK National Measurement System, which is a collaboration of laboratories funded by the government that provides the core measurement infrastructure for the UK. We are involved in collaborative and contract research, and we also have a unique role in that we are the UK's government chemist, where we play a role as a referee analyst which can focus in areas which are involved in regulation, such as food, medicine and chemicals. We provide advice and consultancy on these regulatory affairs to the government. I work specifically within the molecular and cell biology team, where we deal with current and future measurement challenges in areas such as advanced therapeutics, diagnostics, and we deliver this through measurement research, calibration facilities, reference materials and training and consultancy. Our overall aim is safeguarding the quality of public science and also ensuring accurate analytical measurement. I'm going to talk to you today about digital PCR, which is a new technology which has the potential to quantify precisely target nucleic acids. Now I'm going to explain to you what is digital PCR. In this particular example that I will show you here, this is a 1 by 20 microliter quantitative PCR reaction. In this particular reaction we have our template of interest. You split your sample by dilution, in that you perform a limiting dilution where you end up with some reactions containing 0 templates. In this example, you have 20 digital PCR reactions, each of a 1 microliter reaction volume. The PCR is performed as normal using standard real-time PCR chemistry. In this particular example, if you perform absolute quantification of your sample, you count the number of positive or negative reactions. In this instance, that is 10. Poisson statistics are applied to account for multiple targets per partition. Of note is that digital PCR does not require a standard curve as quantitative PCR does. Now I'm going to talk to you about the applications for digital PCR. The ability to be able to accurately quantify nucleic acids is useful in making many types of measurements. One of these is in absolute quantification, which is the example I previously showed to you. And this is useful in areas such as cancer, looking at genetically modified organisms, as well in the field of infectious diseases. Digital PCR allows you to look at copy number variation, as it can be highly precise. This is useful in areas such as cancer, GMO, and in prenatal testing. In digital PCR, you can look at minority targets as well as rare single nucleotide variants. And this has got many applications in different fields, such as in cancer, prenatal testing, organ transplant, and in the field of infectious diseases. Now I want to show you an example of how digital PCR can be used for absolute DNA quantification. In this particular example, this is the results from the quantitative PCR of some samples. So it shows the copies per reaction of this analysis. The black points with the black line is the UV spectrometry estimate copies per reaction of these samples. The blue line is where there is no carrier contained within the qPCR reaction. And we can see good agreement between this estimate of copies per reaction and the UV result estimated copies per reaction. And the red line, we can see there is a difference when the reaction contains carrier. However, when you perform this analysis using digital PCR, we get a different picture. In this instance, we can see that the copies per reaction of the no carrier and carrier are in total agreement with one another. However, the digital PCR is underestimating the copies per reaction as assigned using the UV spectrometer. In this instance, it is worth noting that the UV spec result has been used to perform the standard curve which is being used for the quantitative PCR. So in the instance of the digital PCR, this is free from the standard curve. Now I'm going to talk to you about another example where we are using digital PCR to perform absolute quantification, in this instance in Mycobacterium tuberculosis complex. We have 3 levels of test materials in this example. One is a TB control plasmid, which contains a copy of the 16S ribosomal RNA gene and the RPOB gene. We're also looking at genomic DNA using the Mycobacterium tuberculosis laboratory strain H37RV. Our third set of materials is a BCG material in an artificial sputum matrix, where we are looking at a whole microbe material and relating this to colony forming units. I'm going to talk to you about some of the results from these sets of experiments. With the materials that I showed you, you were able to characterize the samples using digital PCR. So in this instance, if we look at different assay targets and different primer sets for 2 different materials, what we are able to see is that there are, in some cases, a significant difference when using one of the assays on 2 different materials. However, the other assays, there is no significant difference between them in terms of estimating the copies per microliter in that material. Also, we can look at the effect of using different master mixes on our 2 different samples. So in this instance, we can look at a whole cell sample as well as a genomic DNA sample. Again, we can see in some instances there may or may not be significant differences between these master mixes. Being able to use different materials and applying the method of digital PCR allows you to evaluate the differences that these types of materials can make on your estimation of absolute quantity. There are many commercially available digital PCR platforms. Some of these use a prefabricated partitioning, which perform the digital PCR in a plate-based format, like the Fluidigm Biomark and the Thermo Fisher QuantStudio, which feature integrated fluidic circuit technology. These use the digital PCR format to partition individual reactions into microfluidic chambers. The Formulatrix from Constellation and the JN Medsys from Clarity also use a prefabricated partitioning. There are also droplet-based digital PCR systems available in which the PCR is performed in microdroplets separated by oil. The Biorad QX200, the Raindance Raindrop, and the Stilla Technologies Nykeya system all use droplet partitioning for digital PCR. Most of these platforms can generate thousands to tens of thousands of individual reaction volumes. Now we will focus on the 3-color digital PCR platform from Stilla Technologies, and I will show you a short video.
At Stilla Technologies, we have developed the Naica system as the first digital PCR system that combines a simple and fast workflow with a unique 3-color multiplexing capability. At the heart of the Naica system lies the Sapphire chip, our state-of-the-art consumable that integrates the entire digital PCR workflow. Partitioning, PCR amplification, and readout start by removing the white caps on the Sapphire chip and pipette 25 microliter of PCR mix into each inlet port. Then simply seal with the PCR caps and place up to 3 Sapphire chips in the Naica geode, our combined droplet generator and thermal cycler. Close the lid, click play, and walk away. By pressurizing the geode, the mix flows into an oil-filled microchamber engraved within the chip, where it spontaneously breaks up into a large 2D array of 30,000 nanoliter droplets. This regular self-assembled droplet array is what we call a droplet crystal. The geode performs fast PCR amplification of the targeted nucleic acids by thermocycling the crystal in about 1 hour. After PCR, transfer the chips on the Prism 3, our automated 3-color fluorescence imaging system, which scans up to 12 of your samples in just 10 minutes. Finish by analyzing your 3-color digital PCR experiment with our intuitive Crystal Miner software. Positive and negative droplets are automatically identified for all 3 fluorescence channels, the number of droplets are counted in each population, and converted into the absolute concentration of the target DNA sequences. With the Naica system, discover the simplicity of digital PCR.
Now, after watching that video, hopefully you're more aware of how the Stilla platform works. The Nykeya system is based on this unique microfluidic technology, and it allows multiplexing with the unique 3-color detection. It uses a Sapphire chip, which is already pre-filled with oil, and this is very easy to use and you can set up your experiment quickly. All you need to do is pipette your sample with the PCR mix and the primers and probes into the chip and you're ready to go. Then you have the Nykeya geode thermocycler, and this performs the droplet generation and amplification, and the Nykeya Prism 3, and this enables the chips to be read. You can then analyse the chips using the Crystal Miner software. With the Nykeya system, you can use both hydrolysis probes and evergreen chemistries, and you can perform PCR as well as 1-step reverse transcriptase PCR, so therefore you can use both DNA and RNA as a template. The microfluidic channels are able to generate 25 to 30,000 droplets, and these assemble as a 2D monolayer, which is called a droplet crystal. The targets are then amplified, and positive and negative droplets are counted by the software. In this particular example, we can see that counting of the positives in the blue channel are giving 102 copies per microliter, in the green channel 152 copies per microliter, and in the red channel we're just getting 8 copies per microliter. This counting is performed by the software, and this allows you to perform an absolute quantification of your nucleic acids. So briefly, you pipette the PCR mix into the sapphire chip and you seal this with a cap. You place the chip into the geode. Then once the PCR is ended, you put this into the Prism 3, and this is then scanned in 3 different channels: the blue channel for FAM or evergreen, the green channel for Psi3 or hex, and the red channel for Psi5. And then you analyse your data using the crystal miner. From loading the chips to analysis, the whole process takes a minimum of 2.5 hours. It is a very versatile system. Not only can you perform PCR, you can do RT-PCR and whole genome AMP. You can also image the droplets pre-PCR and recover droplets after amplification. Stilla Technologies are working on a 6-colour reader to increase the multiplexing capability of the system. The particular project that I am working on in applying this technology is called AntiMicroBal, and this is a European metrology programme for innovation and research funded project to look at novel materials and methods for detecting, traceable monitoring and evaluating antimicrobial resistance. The consortium which is delivering this AntiMicroBal project is bringing together 11 partners from 5 countries across the EU. It consists of 6 national measurement institutes: LGC, LNE, NIB, NPL, PTB and Tubitac, 1 university, UCL, 3 hospitals, GOSH, UCG, UWH and a stakeholder charity, BSAC. There are also many other stakeholders which are involved in this project, such as other NGOs and societies, different EQA providers such as Charité, Public Health England and Instand, as well as other University College London hospitals and clinical laboratories, and other NMIs, universities and industrial partners. The AntiMicroBal project is aiming to apply innovative metrological concepts to underpin diagnostic analysis and support standardization of antimicrobial resistance testing. It wants to do this through developing reference methods and materials in the areas of routine clinical testing of bacteria and viruses and in point-of-care tests. It wants to do this by supporting external quality assessment schemes and also evaluating new and emerging methods to development recommendations on how these approaches could be applied in a robust and reproducible manner. Ultimately, it is looking at improving clinical measurements to improve treatment choice. Now, in the context of the AntiMicroBal project, I'm going to talk to you about detecting methicillin-resistant Staphylococcus aureus. The organism Staphylococcus aureus is a common skin and nose microorganism. It is usually harmless, but it can cause disease if it enters through a cut in the skin. And this can most commonly present itself as a skin and soft tissue infection, such as a boil. Or it can cause deeper infections where it invades the bloodstream and spreads around the body, causing serious life-threatening infections such as septicemia, meningitis and pneumonia. If the organism Staph aureus is resistant to an antibiotic called methicillin, then this is called methicillin-resistant Staphylococcus aureus, or MRSA. MRSA is of a huge public health concern and medical community concern. Different antibiotics need to be used to treat and prevent MRSA, and these antibiotics are generally more expensive and can have more serious side effects. It's important to reduce the number of staph infection caused by MRSA because it's important to limit the spread of this drug resistance. There are different approaches that can be used to detect MRSA, and some of these use a culture approach. In general, they look at determining growth in presence of the antibiotic. and examples of these are the sulfoxetine disc diffusion method and the latex agglutination test. There are also chromogenic agar which can be used for identifying MRSA, such as the Biomeru Chrome ID MRSA and the BD-BBL Chrome agar MRSA. Molecular approaches in general tend to look at detecting the MeCA gene, which is indicative of methicillin resistance, and these typically use PCR, the polymerase chain reaction. Culture-based methods are in general of a lower running cost compared to molecular methods. They can, however, take more time. Molecular methods have the potential to have a higher sensitivity, but they do require a higher level of skill for the technologist. Molecular methods are available from companies such as BD, and there is an expert assay, as well as from Roche. So this is the experiment that we're doing on the Stilla platform, where we're using MRSA as a model system. As the Stilla is a 3-colour platform, we can use it to target 3 regions of interest: target 1, target 2, and target 3. On the blue, green and red channels respectively, and we are using clinical assays to be run on the platform. From the results, what we're hoping to determine is the sample MRSA, is it MSSA, which is methicillin-sensitive Staph aureus, is it MRSE, which is methicillin-resistant Staphylococcus epidermidis, or is it MSSE, a methicillin-sensitive Staphylococcus epidermidis? Staphylococcus epidermidis is a coagulase negative staph, which can be commonly found again on the skin and in the nose. It can sometimes carry the methicillin resistance, but it is not of as serious a public health concern as MRSA. So it would be useful to determine in a particular sample where exactly the methicillin resistance is coming from. So now to highlight how we are going to tell what we have present in our sample. Here is our bacterial cell, and inside in our bacterial cell we have a bacterial genome. If we perform a PCR of this sample, we're going to look at targeting 3 different regions of interest: target 1, which is blue, target 2, which is green, and target 3, which is red. Now let me take you through how we will identify whether the sample has got methicillin resistance and whether it is Staphylococcus aureus or Staphylococcus epidermidis. In this particular example, we are only getting positive amplification from target 2, which is indicating it is Staphylococcus aureus. But there is no amplification from target 1 and target 3. So in this particular example, we're calling this sample MSSA, so methicillin-sensitive Staphylococcus aureus. So now in this example, the second example, we're getting amplification from target 1 and target 2. Target 1 is indicating positive amplification from MeCA, which indicates methicillin resistance. And we're still getting amplification from target 2, which is telling us it's Staph aureus. So here we have MRSA, methicillin-resistant Staph aureus. So now we go back to our sample again and we perform the PCR. And we get amplification of target 3, indicating in red. Because we have only got red, there is no amplification from any other regions, we can confer that this is methicillin-sensitive Staph epidermidis. So we perform another PCR now on another sample, and we're getting amplification in target 1 and target 3. Target 1 is indicating MeCA, which is methicillin resistant. So here we have a methicillin resistant Staphylococcus epidermidis. So in those 4 scenarios I've spoken to you about, we can determine where the resistance is coming from and whether the organism is Staph aureus or the less serious in terms of public health concern, Staph epidermidis. So now I'm going to show you some of our experimental results. What we're looking at here is where the sample is partitioned into a large self-assembled 2-day array of droplets. And these are called droplet crystals, and this is because the droplets actually self-arrange into hexagonal mesh, and this looks like a crystal when you zoom in. Each droplet contains a multiplex PCR reaction. You can then visualize these droplets in the 3 different channels. In this first example, we're looking in the FAM channel, so the blue channel, and in my example, this is detecting methicillin resistance. Here is a scatter plot, and this is displaying raw fluorescent data from the digital PCR. In this 1D plot, you can see the fluorescence values for the blue channel, and these are expressed as relative fluorescence units, RFU, and these are plotted on the vertical axis, whereas the values displayed on the horizontal axis represent the droplet index. And this is the order in which the droplets have been analysed. You can look at this data from the 3 different channels. So from the green channel, in our instance, this is detecting Staphylococcus aureus, and in the red channel, which in our experiment is looking at Staphylococcus epidermidis. Not alone can you look at this data in a 1D plot, but you can also look at it in a 2D plot. And in this example, you see the intensity values for one of the 3 detection channels, and this is displayed on the vertical axis, and on the horizontal axis you have the fluorescence intensity values for one of the other 2 detection channels. Here we can see we have a population of droplets which are amplifying in each one of the sections. Some which are amplifying solely for Staphylococcus aureus, indicating in the green droplets, some which are just amplifying for the blue, which is indicating Nykeya, and then we are getting a grouping of double positives, which is this light blue colour, and this is showing us some of the samples which are amplifying for both Staphylococcus aureus and Nykeya, so these are MRSA. Now I'm going to show you some more results, which is looking at some samples and the corresponding copies per microliter of those samples. In this example, we have 4 samples that I'm going to show you. And now let's look at sample 1. So on the y-axis, we have copies per microliter, and on the x-axis, it's the sample number. So for sample 1, we're getting amplification of target 1 and target 2. This is methicillin resistant Staph aureus. And if we look at the actual numbers, what we're getting is 59,905 versus 57,901 copies per microliter. If we look at sample 2, we're getting amplification in target 1 and 2 again, MRSA, and as before, I'm also just going to mention that there's no amplification in target 3. In this instance, we're getting 8,922 copies per microliter, and in target 2, 8,751 copies per microliter. For sample 3, again, just amplification for target 1 and 2, and no amplification for 3, so MRSA, and we're getting very close numbers in terms of copies of target 1 and 2, as we saw with sample 1 and 2. We're also looking at sample 4 here, where we have copies of target 1 is 28 and copies of target 2 is 31. So in this instance, what I'm showing you is a range of samples where we're looking at different magnitudes of copies per microliter, and we're getting good agreement with no significant differences in terms of copy number between target 1 and target 2. All of these samples are MRSA with no amplification of target 3. So now we'll look at the next set of results, and again we have copies per microliter on the y-axis and sample number on the x-axis, and in this instance we just have two samples. So if we look at our first sample, what we're getting is amplification in target 1 and in target 3. In this instance, the copies are also very similar in terms of numbers per microliter, with no amplification in target 2, and in our experiment target 2 is Staphylococcus aureus. So here we have a methicillin resistant Staphylococcus epidermidis. And here if we look at sample 2, again we're getting agreement between the copies per microliter for target 1 and target 3, with no amplification from target 2, again indicating that we have MRSA detected. What is this? This is also showing is that the experiment is working in terms of at different levels of magnitude, so it's showing linearity at both high levels of copies agreeing between different assays which are targeting different regions of interest, and also it's agreeing when we have lower amounts of sample, thus indicating that the experiment is behaving in a linear fashion. So another important experiment that we performed on the Stilla platform was to look at the repeatability of the data that we were obtaining. And so we did this in the first instance looking at MRSA. So here we have a comparison between different chips. All the samples have been performed in the same run, but what we are getting is no significant differences in terms of copy numbers per microliter, in this instance when looking at target 1 and at target 2. So the dots are indicating the copies per microliter for each target, the blue circles are target 1 and the green squares are target 2, and the black line in the middle is the average value. And we also did the same experiment for methicillin resistant Staph epidermidis, and here what we're getting as well is no significant difference in terms of copies per microliter between the different chips and between the replicate samples. In this particular graph we can see that the red triangles indicate target 3 and the blue circles are target 1. This is showing good repeatability of the data with CV values of less than 4%, and this was observed for all the assays and for all the materials that we were using. So to follow on from the experimental results that I have showed you on the Stilla platform, where we have seen very good linearity and very good repeatability of the data, we would then like to move on to a more complex model where we would analyse blinded clinical samples. And in this instance, we want to determine, can we confirm the clinical result? Using our particular assays, we can look for if there is MRSA there or not, or if methicillin resistance is actually coming from the Staphylococcus epidermidis, or if there is just methicillin sensitive Staphylococcus epidermidis or Aureus in the sample. We would like to investigate the performance of the Stilla platform in this particular experiment. We'd also like to look at further applications of the Stilla platform. In the particular AntiMicrobial resist project, it's to look at further models for antibiotic resistance determination, as well as in the areas of applying this platform to precision medicine and synthetic biology. I'd like to acknowledge the many people involved in this work. So at LGC we have Jim, Rebecca, Alexandra, Eloise, Carol and Helen. At Helicus Clinicum there's Benyam and Parvis. At Stilla Technologies, Magali, Caroline, Sarah, Alexandra and Laura. And at University College London we have Tim and Emmanuel. And at Great Ormond Street Hospital there's Catherine. Also we've got our funding from Bayes, and for the AntiMicroBial resist project, all of the partners that are involved in this project, as well as MPIR for the funding of the project. I would just like to say thank you very much for listening, and I'm happy to take any questions that you may have.
Thank you for your presentation, Denise. It is now time for the Q&A. This is your opportunity to ask not only Denise your questions, but also Magali Druno, Director of Applications at Stilla Technologies, who is here to answer any technical questions you may have. To ask a question, all you have to do is type it in where it says ask a question and then press submit. So our first question asks, why are you performing digital PCR? Could you just do the same type of detection using quantitative or real-time PCR, Denise?
Thank you for that question, Jason. So really, you were applying the technique of digital PCR when you want to more precisely count the number of targets in your sample. So it allows you to perform this without the standard curve, which is what is required for qPCR. So in that instance, you get an actual independent measurement of your material rather than having to relate it to a standard. It also has been spoken about in having an improved accuracy even when there are inhibitors present in your sample. And it can be more forgiving when you have a poor amplification efficiency in your PCR.
Excellent. Thanks, Denise. And our next question asks, you mentioned the term limiting dilution during your presentation. What does that mean?
So really what it means is you need to dilute down the target in your sample in order to partition it into the individual digital PCR reactions. So that's really what it means, is you are diluting down that target in order to get either 1 or 0 targets in that reaction.
Excellent. Thanks, Denise. And our next question asks, do you see fluorescent spill over? And if so, how do you control for it?
So because it's the Stilla platform has that 3-color system, you perform what's called a color compensation experiment in the first instance, and this is where you run your individual target with all of the assays, and then the software allows you to apply that color compensation, which corrects for that spillover that you may see between the different channels, and you apply this then when you run your particular experiment. I'm not sure if Magali, if you would like to add anything to this question in terms of the fluorescent spillover, or anything you would like to add?
I think you said it all.
Okay.
Basically, so fluorescent spillover, that is the detection of interfering signal from fluorophores that should be detected in adjacent channels. So we can correct for it, and we have a system where you just have to set it up for your assay once, then you can just apply it for all your subsequent experiments. So this is quite an easy feature to use, and most of our users are quite satisfied that they can adequately compensate for the assay.
Excellent. Thank you both for that answer. Our next question asks, is it possible to sort of access the raw data from the Naica system? So, for example, so that you can check the sort of acquisition parameters. Magali, is that something that perhaps you can answer?
Yes, you can actually access the raw data, so that means an image of your droplet crystal, and we've got a set of tools so you can actually make sure that you are not overexposing your droplet crystal, that the basically the time of exposure is not too long, and that all other acquisition parameters such as focus are correct. You've got also little quality flags to help you decide whether this is okay or not.
Excellent. Thanks, Magali. And just a quick reminder to our live audience, you still have time to ask a question. All you have to do is type it in where it says ask a question and then press send. Our next question asks, do you have results from mixed strain samples? Denise, is that something that you can perhaps answer?
Yeah, so this is actually a very good question, and it's very difficult to know how. So we can set this up, since in the lab we can do a mock-up experiment. What's difficult to know is how clinically relevant a mixed sample is. What seems to be worried about clinically is the methicillin resistance there, and if it's coming from the Staph aureus or the Staph epidermidis, because one would have a greater public health concern. So really, this is not really fully resolved. We need to test clinical samples to really understand, are we able to pick up when there are mixed samples? All we can do at the moment is we've kind of done mixtures. So as I've said, kind of more with further work, hopefully we will have more on this topic.
Excellent. Thanks, Denise. And our next question asks, in your experience, is setting the fluorescent threshold between the positive and negative droplets challenging?
No, not when you can. So it's actually quite simple. We found now with our assays, I guess it may depend how you develop and optimize your assays, but in these particular assays we were using, they were developed on real-time PCR, and they directly transferred. There was some optimization in terms of number of cycles, which gave a greater discrimination between the positive and negative droplets. But we didn't see any issues in terms of setting thresholds.
Excellent, thanks, Denise. And our next question asks, can you use digital PCR using the Naica system to quantify, for example, circulating tumor DNA? Is that something that's possible?
Yeah, absolutely. And this is actually where quite a bit of work has been done, because the accurate detection of circulating tumor DNA in plasma is very challenging because the amounts can be very low. So actually, digital PCR can facilitate that, detecting those low amounts. And it is being applied certainly on a research basis, and probably looks very promising to be applied clinically in what's called looking at these liquid biopsies, which are these trying to detect these mutations in blood samples from cancer patients, for instance.
If I may ask, actually there is a recent publication that been just out in the last month in PLOS ONE, actually detailing the use of the Naica system for the quantification of circulating tumor DNA in lung cancer patients. So I just invite everybody to go and read it.
Excellent, thanks Magali, and thanks Denise. Our next question asks, did you use pre-purified DNA sample or an unpurified DNA sample in your project?
So for this was just DNA extracted from culture. We didn't apply any further purification for the DNA.
Excellent. So there's really sort of no sort of sample prep needed before sort of carrying out the digital PCR using this system?
Not in this instance. I mean, what could happen when we move to directly analysing clinical samples, we may get some presence of inhibitors, but as I was mentioning, digital PCR is normally quite forgiving of that, but certainly in this instance of where we were just using a culture, simple enough culture, single organism approach, we didn't perform any further purification.
Excellent, thanks Denise. And our next question asks, how long does it sort of generally take to get the results using this system? How long did it sort of take you?
So in about 3 hours, but that's the minimum, that's really the minimum time I would think, but that's not including maybe the extraction of your sample, which would be on top of that. But from your actual DNA to your result, that would be approximately the time.
Excellent, thanks Denise. And our next question asks, do you need to analyse several dilutions of an unknown sample to achieve the limiting dilution? So this is referring back to when you were talking about the limiting dilution.
Yeah, so some of the digital platforms have a narrow dynamic range, but actually the developments that have happened mean you have to worry about this less. But it definitely, if you have an unknown sample, you could run the risk of totally maxing out your droplets, because you have to have some negatives in order to be able to quant that sample. So definitely you might like to run a sample neat and a 1 in 10 on the first instance, or you might just like to run it neat and then kind of get a rough estimate. For instance, if you're saturating your the fluorescence in the detection, then maybe you would look at then performing some dilutions of that sample.
Excellent, thanks Denise. And our next question asks, how much is the maximum copy number tolerance using this system?
So in our instance, we were looking at about a range between 10 and 600,000 copies. You have to so probably if we were to go above that, if we were to go to probably over 1 million copies, we would run the risk, or even more than that, we would run the risk of probably saturating the reaction. Yeah, so hopefully that has answered that. So what you need to do is you apply, when you dilute that sample, you then apply that dilution factor to calculate what you have in your original sample.
So just to complement indeed on Denise's answer, dynamic range is actually 0.2 copies per microliter to 20,000 copies per microliter. But that's the final concentration, so that's in the mix. So indeed, as Denise was saying, then you can input, because we give the result as stock concentration, if you multiply whatever concentration you add by your dilution factor.
Excellent. Thank you both for your answer there. In our next question asks, in your presentation you refer to copies per microliter. So what concentration were you actually referring to?
So that's the copies of your target per microliter of your extract. So your DNA, for instance, if you have 100 microliters of your sample, that's what I would be referring to, per microliter of that sample.
Excellent, thanks Denise. And our next question asks, can the method that you've described work on other digital PCR platforms?
Yes, but currently they are limited by having 2 detection channels, whereas the Stilla platform does have that 3-color detection system. But you can do something, you might be able to do something where you can do almost like differential amplification. So it's where you might like have different temperatures. So at certain temperature, you may get your amplicon appearing, or your positive droplet population may appear at a certain amplitude versus maybe at another temperature it may appear at a different amplitude, but you're talking about a more complicated experiment in that instance. But what the Stilla affords you to be able to do is actually to have a channel per target.
Excellent, thanks Denise. And our next question, not too sure if you can answer this one, but poses the question, if you have two DNAs which represent each of your target candidates, and they happen to be co-encapsulated into a single droplet, that's going to cause some sort of problem. How can you overcome that using this sort of method?
So yeah, that can happen. You could dilute your sample. You could in digital PCR, what tends to happen is you apply something called the Poisson statistic, which calculates the probability of this particular event happening, and this allows you then to convert that count into an actual quantification of your original sample. I'm not sure, Magali, if you'd like to add anything for that question.
No, I think you answered it very well.
Okay.
Excellent, thank you. And our next question is sort of a general question, whether you can sort of state the difference between a droplet PCR and crystal digital PCR. Magali, perhaps that might be something that you might be able to answer, or?
Sure. The main differences stem first on the way the droplets are generated. We generate our droplets on chip, and they remain there as a 2D monolayer array, whereas in the Biorad droplet PCR system, the emulsion is formed by another microfluidic technology. They will be collected as a 3D in a tube, and then basically thermocycled before reading one by one. So in a process akin to flow cytometry. Whereas what we do is that we've got a monolayer of droplets that remains on chip. You just place the chip onto the thermocycler, a flat block thermocycler, and then you acquire an image, and not just a fluorescent signal. So you acquire an image of your droplet crystal, which you can just then visualize. So it's droplet-based, but it's a different way of reading it. Also differences, as Denise has mentioned, is the fact that we've got 3-color detection, and another thing is that we do basically allow access to our users to all of the raw data. So you can actually see the image, and you can also see how the software has processed your data. So I would say that these would be the main differences between the two techniques.
Excellent, thank you. Well, I'm afraid that is all we have time for today. Before you leave us, we would really appreciate if you can spare a few seconds to fill in a very quick survey on your experience today. I would now like to thank Denise O'Sullivan for her presentation, and both Denise and Magali Druno for answering your questions. I would also like to thank the webcast sponsors Stilla Technologies, and of course you, the audience, for taking the time to be with us today. Remember, you can watch this webcast again at any time on demand at nature.com webcasts. Thanks for watching, and I hope you'll join us again soon.